Okay, folks. Well, it's a treat on the Caris Molecular Minute podcast to have Dr. David Spetzler with me again. David, it's been about a couple of years since I've had you in the podcast, so you got to play your cards much better so I could bring you more often.
I'll do my best.
Yeah. So probably most folks who are listening and viewing this know a little bit about you, but would be nice just to refresh folks just to who is David Spetzler?
Yeah, so I'm a long-term employee of Caris. I've been here almost 15 years now. I'm a mathematician and molecular cellular biologist by training. I was the very first bioinformatics scientist hired by Caris, and now I'm the president. So,
And you were at the age of 10. You started at the age of 10.
yeah, I just age well, I guess. I think I'm older than you are, Chadi.
Yeah, no, I'm probably older. But David, one of the things that are within your purview in the company is product development and strategic thinking into what's next for Caris in terms of products in the era of precision oncology or precision medicine, frankly, broadly. And one of the things I want to focus on today on today's podcast is liquid biopsy. So why should we care about even having a liquid biopsy out there scientifically?
That's a loaded question. So liquid biopsy creates opportunities that just don't exist with tissue. So at the highest level, it's a non-invasive test, right? So you can only biopsy someone so many times. There are significant risks associated with biopsies, even mortality risks. So being able to understand the evolutionary response of cancer to therapeutic pressure can only really be done in an ongoing basis through a liquid biopsy. So if you want serial monitoring, serial measurements, it has to be through a liquid biopsy.
And over the past couple of years, I know a lot of you guys in the lab have been working heavily on bringing our Caris liquid biopsy to the public, namely Caris Assure. What is Caris Assure?
So Caris Assure is a whole exome, whole transcriptome, chip-corrected approach to liquid biopsy. And so what we're doing is we're extracting cell-free DNA, cell-free RNA from the plasma, and we're getting the genomic DNA and genomic RNA from the buffy coat and sequencing both of them, all coding regions for all genes. And what we're able to do with that then is identify mutations that are occurring in the plasma that are not in the buffy coat or at much, much lower variant frequency in the buffy coat. And that's really, really important because we're finding that about 30% of patients have a chip alteration in a cancer-related gene. And so that leads to an incredibly high false positive rate. It's not a technical false positive. The mutations are actually there, but it's a clinical false positive. Because what you don't want to do is, for example, treat a prostate cancer patient with a PARP inhibitor because you find a BRCA2 mutation if that BRCA2 mutation is not coming from the tumor, right? So if it's a chip alteration and that BRCA2 mutation only exists within the white blood cells, then that's going to be an ineffective therapeutic intervention. And that chip correction is of incredible importance. And what we've found is that that 33% rate is pretty consistent in terms of then that false positive rate of other assays that do not do the chip correction. And so we've been able to achieve much, much higher performance metrics by using this dual approach where we're measuring both the plasma and the buffy coat. And so we're sitting at PPVs that are in the high 90s compared to existing plasma-only assays where the PPV is down in the 60s and 70s. And that has a very meaningful impact on patients to have such a high false positive rate.
So, so there's a lot of information here. To summarize, basically, because we're sequencing the buffy coat, we're able to tell if a mutation is coming from the tumor versus from white cells.
That's correct. Yeah.
Yeah. And are others looking at CHIP? Are others sequencing the buffy coat, to your knowledge?
So I don't know that there's any clinical assay. I'm unaware of any clinical assay besides ours that is doing that buffy coat correction. Certainly, there's been some really good work out of many academic labs showing how this is necessary. There's a lot in the literature there. And in fact, the initial work showing how there are a lot of DNA repair enzyme alterations that are of CHIP origin. That came out of work by Colin Pritchard. So this is not something that we discovered. It's something that I think has been established in the literature. We're just implementing it at a clinical scale because it's too important to us not to do the best science we possibly can.
I have to tell you, David, I was a little bit surprised. I mean, as I studied a lot about CHIP, I did not realize that it could be up to 1/3. Like I had the impression it's much, much less. And as we learn more, we're seeing a lot of these that are really false positives.
That's right. Well, people assume that, okay, when you're talking about a CHIP mutation, you're talking about a mutation in DMNT3C or TET2 or one of the common CHIP genes. And of course, the frequency of those is much, much higher. Those aren't the problem. Nobody's going to be making a therapeutic decision based upon those genes. The problem is when you find a KRAS CHIP. We found KRAS G12D and KRAS G13D mutations that are CHIP of origin. We found BRAF V600E mutations that are of CHIP origin. Those can have a profound effect on how patients are treated. And of course, are very important. BRCA2 we find quite significantly mutated and is coming from a CHIP origin. So I think that there's been so little work done in this area in terms of broad-based testing in oncology that we don't know how big a problem it is. But that's where this approach becomes so important, is that we're beginning to accumulate data on thousands and thousands of patients to start to understand the frequency of these CHIP mutations that look like oncogenic drivers but are not. And that's going to be incredibly important to make people aware of.
Yeah, and really, I think you hit the nail on the head, because it's not the ones that don't have a treatment options. It's the ones that could potentially have a drug, and then you use the drug inappropriately. You could get the patient side effects that they don't need. The cost of care goes high, and it's a whole slew of things that probably we can avoid.
That's right. Or withholding a drug.
Right. Yeah. So I mean, at Caris, we've been doing whole exome, whole transcriptome sequencing on tissue for several years. And obviously, this is a big differentiator for us in the lab and as a company. It helps a lot of our researchers as well as our clinicians. When you look at the liquid biopsy, how do you compare and contrast between liquid, Caris Assure, and what we're doing in tissue?
Yeah, so the same reason to do it in tissue is the reason to do it in blood, because we don't know what we don't know. And the only way we're going to find it is if we look broadly. And it has very, very practical implications, for the patients of today. And a perfect example of this is TMB, right? So when you look at total mutational burden in the blood, the correlations back to tissue have been really quite bad. And if you want to have a reasonable specificity, then sensitivities are sitting down in the low 30%. And that's still with not a super high specificity. And so by going to whole exome, right, we're measuring so much of the coding region, I mean all the coding region essentially, that we're able to find a lot of these mutations. And then when you do the chip subtraction, you're not being unduly influenced by an age-related phenomenon in terms of the accumulation of these mutations. And so our performance on TMB is absolutely outstanding. I mean, we're talking about north of 90% sensitivity with 100% PPV. And that can only be accomplished by doing whole exome and by having the chip subtraction. And so, when we start to look at these genomic features that are, genome-wide, or at least exome-wide, the way we do TMB, there's a huge advantage to measuring, all genes. And the same is going to be true for LOH and, for all these structural components that are emerging as potential targets.
Have we, I mean, I think we can both agree that tissue will always remain the best possible option, although you can't, like to your earlier point when we first started, you can't really keep biopsying. It's easier to get a tube of blood. But how have you seen the concordance between Caris Assure and the tissue?
So we can break it up into two different groups of mutations, and I think that's important to do. So when we look at the driver mutations, those truncal mutations that are the cause of the cancer, we're looking at, again, greater than 90% sensitivity, 93.8% sensitivity in our validation study. And that's with very, very high PPV, 94% PPV. That performance doesn't hold true when you start to go to the passenger mutations, right? So if it's a low variant frequency part of the tumor and it's only in a small subset of the tumor clones, then of course it's more difficult to detect in the plasma as well. And so, most of the things that treatment decisions are being made off of are the truncal mutations, are the driver mutations, right? You got to find that EGFR positive patient. You're looking for KRAS and PIK3CA. And so, but of course you want to pick up everything that's possibly there. And so when we, expand it to the passengers, definitely the concordance drops down, still phenomenally good, I would say, at, closer to 70%, 75% sensitivity. But it's accounting for that, tumor heterogeneity, and the fact that a lot of these are more rare. And the other thing that I think becomes telling with that is that we can see a decrease in that correlation between tissue and blood being affected by the time difference. So how quickly was the blood collected after tissue or tissue after blood? The closer they are in time, the higher the concordance. But if you wait 3, 6, 9, 12 months, then that concordance goes down. And I think that's a reflection of the change in biology of the tumor. I mean, these are metastatic cancer patients. They're going to go under treatment for the most part. And so their tumor will be changing and evolving in response to therapeutic pressure. And so I think, when you do a direct comparison to tissue, you treat those as false positives and false negatives. But a lot of that is true, biological changes in the nature of the cancer.
And I want to really hone on that point for those who are listening and viewing this, because that's really important. It's a nuance, it's a small point, but it's so critical, because you get the tissue and then patients receive a lot of these treatments, and the impact of treatments on the mutations that you may detect in blood are huge. It's almost like when you have to re-biopsy a patient after a year of treatment because you don't know what may have happened during that year because of therapy. It's really a nuance, but it's so critical.
It's part of the power of a liquid biopsy too, right? So, as the data emerges and we move from kind of a single time point for therapy selection to multiple time points for disease monitoring and therapeutic response monitoring, we're going to start to see these resistant clones emerge in the blood. And the sooner we know about that, the better informed we can be in terms of modifying therapeutic strategies to account for those resistant mechanisms. So there's going to be a tremendous amount we can learn from liquid biopsies. So
we just agreed that you've been living in that space since you were 10 years old. Someone out there maybe listening and say, so many products out there, a lot of, the field is crowded, so many companies having these liquid biopsies. So I guess what I would like to learn from you is, where do you see Caris Assure as different than other products that currently exist, in your opinion, as you lived in that field for many years?
Sure, there are two giant differences. The first, of course, is chip subtraction that we've already talked about. Absolutely essential to avoid false positives. But the second is in the number of genes. So when we think about, cancer, and we think about evolutionary biology, is cancer going to limit itself to, 80 genes or 200 genes or 400 genes? No way. The cancer will use every available mechanism to avoid death through the therapeutic intervention. And so we don't know about a lot of the other resistant mechanisms because we haven't been measuring for it. If we don't test for it, then we can't find it. And so the only way we're truly going to start to make inroads into understanding all the different resistant mechanisms that can emerge is by doing very, very broad profiling. It's got to be broad and it's got to be deep. And there is no other assay that I've ever seen that comes close to the breadth and depth of Assure. And that's, put that on top of the chip correction. It's phenomenally powerful.
And a lot of the genes that today may not be relevant, with time, we may discover that they are relevant. KRAS 20 years ago, nobody thought has anything to do. I mean, we knew it existed, we knew it was bad, but we never thought it was going to have a treatment.
I mean, in 2015, MSI was for Lynch syndrome.
Yeah.
2016, now all of a sudden, it's an immunotherapy predictor. And we don't know what is going to happen in terms of therapeutic development. And so the only way you prepare for the future is by measuring everything. And then whatever new drug comes out on the market, we're covered.
So if I ask you, what does Caris Assure bring to the field of precision medicine or precision oncology, what I'm hearing is it brings more precision to precision medicine, because you're really adding more layer of accuracy.
It's even bigger than that, right? So when you have the ability to detect individual mutations with a high concordance back to tissue, what it means is that you're measuring everything. And so that initial clinical indication is for therapy selection in late-stage cancer patients. But the exact same assay is what we're using in our MRD study for minimal residual disease, right? When you're measuring everything, then we don't need to change the assay based upon that clinical context. The nature of the interpretation is changing. The clinical context of the patient drives what we do with the information, but it's the exact same assay that is being used for therapy selection, is being used in our MRD studies, and is being used in our early detection of cancer studies. And as you start to think about it, disease are cells going wrong. And so whether we're talking about cancer or rheumatoid arthritis or whatever you want to pick, there is something wrong with cells, and therefore there's something wrong with the molecular system. And an assay like this is going to be able to start to find those drivers and those patterns. And so in many ways, the way we need to be thinking about this is as a universal diagnostic, where once we gather enough data and once we make enough measurements, we're going to be able to find patterns that are, very informative across a wide variety of chronic illnesses, not just cancer.
Yeah, it's almost, I mean, one of my questions to you was along the lines of strategy. When you have something like this that is available at your disposal as a scientist, and you think about what could be done with this, it's hard not to ask the question strategically, how do you take this and position it in the field of medicine in general beyond oncology?
Yeah, I mean, we're going to step into it. So we are running right now a 500 patient MRD study where we have 3 different time points that we've collected plasma on. And so that's a great stepping stone, going from kind of that late stage now into earlier mid stage. And then we've got another multi-cancer early detection study that we're starting, that we've started, where we have 1,600 patients across a wide variety of cancer that are all early stage 1. And then we've enrolled about 15,000 patients in prospective collections for both breast cancer and colorectal cancer. In colorectal cancer, it's a true screening population, all patients scheduled for colonoscopy. In breast cancer, it's a at-risk, a high-risk cohort. It's all women scheduled for breast biopsy. And in that way, we can take this one assay that we've built and begin to characterize its utility across a wide spectrum of clinical uses and indications. But from a technical perspective, it's all exactly the same assay. It's just, what do we do with the data and how do we interpret the results that changes?
David, we did not touch base much on, unless I miss, about germline. And I know one of the things that we've talked about is the ability to potentially capture some of the incidental germline mutations that we could find. I wonder if you could comment on this. I recognize that, this may require further validation and things like that, but I'd like listeners and viewers to understand a little bit of that capability as well.
Yeah, so when you're measuring the Buffy coat, we're finding those germline events, right? So if it's a change in the coding region, then we're finding it. There are certain types of germline events that NextGen is just not good at catching. So, very, very large deletions in PMS2, for example. We're not covering all the promoter regions. So there can be mutations, germline mutations in promoter regions that we won't get, but the current assay is covering north of 99% of germline alterations. And if it's in an AMC G advised gene, then we're going to report it out as an incidental germline finding. So when we find something, we know that it's there, but when we don't find something, then we can't be sure there isn't something else that is not covered by the assay. So that's why.
And that's important. So if you find something, we're pretty certain it's there, and we may recommend that further testing is needed. I think what I'm hearing, and correct me if I'm wrong, is that if somebody is looking for germline specifically, this is not the assay, because if you don't find something, you cannot exclude the presence of a germline mutation.
Correct.
Right.
But we will report those that we find. And, there are weird things that you see. So mosaicism, for example, common in P53. So if you see a 20% variant frequency P53 mutation, it's really hard to know, is that mosaicism? Is that chip alteration of very high frequency? Is it even a germline and you just had a very low read? And so, in those situations, then you really need to go and test fibroblasts really to start to gain insight and start to test different compartments. Because if it's mosaicism, then you're going to find it in some parts of the body and not others. So the complexity of looking at liquid and having more than just the tissue really is quite significant. And so we do our best to stratify, to the best of our ability, the origin of the mutations that we're identifying. So we will absolutely report out that P53 mutation, but we'll say, hey, this is potential mosaicism, and we advise additional testing of primarily fibroblasts to confirm the origin of this particular mutation.
Couple of more questions and then I'll let you go. But one is, you alluded to a little bit, and I want to spend maybe a couple of minutes on it. I mean, as you develop the assay, I'm sure all in our minds, we are thinking some clinical research studies and various studies to take that to the next step. Maybe highlight several of the specific studies that you're excited about where you can actually apply this assay on a particular protocol or a program.
Yeah, I mean, for me, I think that some of the most exciting studies that we have coming down the pipeline are looking at, in the MRD space, expanding beyond what's already been shown to be useful. Right, so some of the most common cancers are now covered for MRD, but that leaves a lot of patients, most patients, without anything. And so getting kind of a pan-tumor MRD assessment, I think, is really, really valuable. monitoring as well. So understanding, when resistance is occurring and figuring out if we intervene then, can we stop it earlier? I think that there are a lot of interventional trials that are going to be needed to be performed to understand what to do with this information. And I'm particularly excited about a lot of those because I think we're going to be able to improve how we treat patients, minimize, overdue toxicity, and extend life significantly. So there's a huge number of interventional trials that can be liquid biopsy guided that need to be done to understand how to use this information most effectively. And it's going to be great to see those done.
One of the most important ones potentially is continuing therapy for some patients who are MRD positive, but also withholding treatment. If you can avoid treatment with somebody who is MRD negative, you're doing that individual a huge favor.
That's right.
So my last question is, like any new product that is coming to market and is going to be utilized, there are obviously some changes, refinements, things that everybody is working on to make sure that our external stakeholders, the investigators, researchers, and really most importantly, the patients who really need this are getting the best of the best. So what should they expect, I would say, over the next year or so as you continue to work on Caris Assure to make it even better than it currently is?
That's a great question. So I think from a performance standpoint, I think we're bumping up against the edge of biology, that we're finding everything that is there to be found. I think we can do better in terms of delivering it quickly. So turnaround time is of utmost importance in these situations. And theoretically with blood, you should be able to get down into that 5-day time frame. I'd like to see us get to that. Already the convenience factor is huge. I mean, the fact that we'll send a mobile phlebotomist to the patient's house to draw the blood there, that's massively convenient for patients and physicians alike, and saving them time and saving them that extra work will have its own benefits. But then getting those results back ASAP, that's probably one of the biggest areas of improvement we can do. We're not bad now, sitting at like 9-10 days, but we can do better.
I know we always have aspirations to exceed our own expectations, which is really what's unique about what we're doing at Caris. And one of the things I actually enjoy usually is what we try to do with the clinical report. The oncologist who receives the report, one of the biggest complaints you hear about when you talk to medical oncologists, I get this report and it's really confusing. I mean, they're not all trained molecular biologists like you. So we really do take extra effort to make this report much easier, including the liquid report that we continue to work on. So I don't know if you want to comment on that, but since I sit on that committee, I really enjoy listening to how we're trying to change things to make it very easy to understand and interpret.
Yeah, I mean, at the end of the day, the first use for this is therapy selection. So that's got to be laid out in very clear, concise, simple terms on page 1. And that's incredibly important to us. Since we're doing such broad testing, the opportunity to provide more information is significant. And so, the other big piece of the report that I think is important to advance the science and the medicine is kind of that additional results summary where we have things like the eukaryotype and where we can put some of the emerging signatures that are on there, CMS4 subtypes, CMS subtypes. There's an opportunity to provide kind of information on the nature of the cancer that won't be useful to everyone, but will be important in the future, and disseminating that early on and getting physicians exposure to it, even if it's in just that research context, is an opportunity for us to help advance the science. And so I think it's it's the 1-2 punch, right? Simple, easy interpretations for, the clinical use today for the patient in front of them, but also having all of that extra information that we can start to glean from the exome and transcriptome that help characterize cancers and do a better job of molecular defining the nature of them. So it's balance.
Is there any question I should have asked you about Caris Assure, strategy, future, anything I may have overlooked and should have asked and I just completely did not ask it?
No, I think you covered everything. I mean, it's a pretty broad topic. It's an exciting time, right? This represents an innovation and a technical advancement that is going to create great opportunities. And so the way I like to think about it is, if you look at what was probably the most important single invention that increases human longevity and human life, I would argue it's the microscope. And that's because the microscope led to the discovery of germs, which led to antibiotics, and antibiotics has had a more profound effect on human health than anything else. And so I think what we have now is a new molecular microscope, and we are going to make discoveries and we are going to find things, and that's going to lead to new therapeutic interventions that will have a very profound effect on human life. And I'm just super excited to play a small role in that journey. It's an exciting time to be alive.
I love this new molecular microscope. I love the idea of molecular microscope. I'm going to use that, and I'll consider attributing that word to you, or I may just steal it completely.
All right, you can steal it completely. Although I guess now it's documented that I told you about it.
Yeah. David Spetzler, President of Caris Life Sciences, thank you so much for sitting with me and talking about Caris Assure, the future, strategy, trials. Very exciting times for all of us at Caris. Really appreciate your time.
Thank you, Chadi. It's always a pleasure.